Hi, my name is Amanda, and in this project, we are doing an investigation of molecular mechanisms associated with sexual dimorphism in sepsis. So, sepsis is a life-threatening organ dysfunction caused by a dysregulated host immune response to infection. But septic shock is a progression of sepsis which is characterized by a circulatory, cellular, and metabolic abnormalities, and is also associated with an increased risk of mortality. So this study published in the Lancet showed that in 2017, almost 49 million cases and 11 million deaths were associated with sepsis, which represents 20% of global deaths. This study also discussed that there is a demographic impact on people considering sex and age, and they showed that the global incidence of sepsis is increased in women, while global mortality of sepsis is increased in men. So in this study, we aim to understand the molecular signatures that may reflect on these sexual dimorphisms in sepsis. So our methods include download the gene expression data of sepsis-related studies and processes this data in R through a pipeline. So our pipeline includes data download, import the data, quality control, normalization, gene annotation, probe summarization, and outliers identification. Next, we do the sex imputation. We perform the differently expressed gene analysis to find also the consistently gene expressed in female and male. We do the deconvolution analysis to find the proportion of cell lines. We perform the co-expression genes. We perform the pathway enrichment, and we also, this is a future step, but we are also interested in gene prediction. So as our first results, we have here a bar graph showing the number of samples for each data sets that we analyzed. So we have the following classes: healthy, sepsis, and septic shock. And we also divided the studies according to age. So pre-puberty represents the neonates and children, and post-puberty represents the adults and elderly people samples. So we have here the number of samples of each study. But we also have a Sankey plot showing the total number of samples, which are divided into female and male, and we have for each class that we analyzed here the number of samples that we analyzed. So next, we performed a consistently gene expression analysis to find those genes that that have more impact on those groups for female and male. So we count the number of genes expressed both up and down regulated, and we consider a consistently expressed gene if they appeared in 6 of 7 studies. So we have here the number of consistently genes in red for upregulated and blue for downregulated. Next, we have here the Venn diagram showing the overlap of genes. So we have some genes related to neutrophils, and they appeared only expressed in female groups. But we also have CD14, which is a gene expressed, a marker for monocytes, expressed only in children's in female group. And we have also other genes here such as MMP8, GPR84, and so on. So we are investigating deeply those genes, and we have on the right the log two-fold change of those genes here. We performed the deconvolution analysis in order to find the proportion of subgroups of neutrophils. So we have here in the heat map the number of studies showing the consistently proportion of those subgroups of neutrophils, and we found that myelocytes and band neutrophils were more increased in pre-puberty samples, but they decreased in post-puberty samples. And we also found that segmented neutrophils were more induced only in male with septic shock, and then they decreased with age. We also found some other cell lines induced only in female. So on the right we have here a bar graph, and we can see that the green bar shows the number of monocytes, and we have monocytes and also CD8 induced in the female group. We also performed a network analysis to find the co-expressed genes more induced in each group, and we found for female were more pre-puberty female have more genes co-expressed in this group. So that's it. Thank you very much, and I'll see you in Lion.